Hello, I'm Tim Weaving, a PhD student at University College London, and I'm pleased to welcome you all to the 16th instalment in CompBiomed's e-seminar series in collaboration with the Virtual Physiological Human Institute, or VPHI. The talk today will be given by Roberto Fino, a computational medicinal chemist at Acellera Labs. Roberto graduated from the University of Bari in 2014 with a degree in organic chemistry, after which he joined the University of Salerno as a research fellow, where he worked towards improved docking and scoring of compounds in high-throughput virtual screening. In 2016, he was selected as an MSCA fellow for the Aegis ITN Consortium and completed his PhD at the Technical University Munich. His thesis looked to combine NMR fragment-based screening, computer-aided drug design, and multi-component reactions in early-stage drug design. He has also been a visiting researcher at the ETH Mod Lab Group and the University Medical Center Groningen. Finally, Roberto joined Acellera Labs in 2020 and works on delivering cutting-edge tools for computational drug discovery. The title of Roberto's talk is PlayMolecule for Drug Discovery, in which he will introduce us to the platform and its role within the pharmaceutical industry, providing enhanced methods of high-throughput virtual screening, computational hit finding, and molecular dynamic simulations applied to biologic systems. There will also be a live demonstration to give us an idea of what PlayMolecule can do. And before we begin, I'd like to briefly remind you that the session is being recorded and will be available on the CompBiomed website and YouTube channel shortly. So please do use this to go back over parts of today's talk and others in the series, and then maybe even share these e-seminars with your friends and colleagues. There will be a Q&A session at the end of the talk, so please save your questions until then. Once the talk is finished, you can post your questions in the questions tab, and I will read them out to Roberto to keep the session running smoothly. Now that's everything from me. I hope you enjoyed today's talk, and without further ado, I'll hand over to you, Roberto.
So thank you, Tim, for the nice introduction. And so today I'm going to show you what PlayMolecule is and how it can be used in workflow to design workflows and perform new discoveries in drug discovery. In a drug discovery context. So first of all, a quick introduction on Acellera. So we are a small medium enterprise based in Barcelona, and it was founded in 2006. So we are almost close to our 15th anniversary. So we have customers worldwide in Japan, Asia, and Europe. And our main focus is to deliver cutting-edge technology based on GPU accelerated calculations, cloud computing, and AI or machine learning. So we focus on computerized drug discovery, and we have developed several tools in order to boost and to improve the computational drug discovery hit finding using machine learning. So we have several collaboration with top-notch pharmas and even with academic lab. So this is a brief timeline of the achievement accomplished by Acellera. So since its foundation, we released AMD, which is our MD engine for molecular dynamics. So this is based on GPU calculation. And then we improved the MD for drug discovery. In 2013, we launched our own workstation for the hardware named Metro Cubo, and which is equipped with 4 GPUs to improve calculations and to speed up the calculation on MD. Then in 2014, we started with adaptive sampling. We launched Sales Cloud in 2015, and then in 2016 we Stefan Dörr, our main developer, developed HTMD, and HTMD is a Python framework to interface to ASMD and other Acellera-based tools. Then we started working on cryptid pocket finding using mixed and solvent MD. And in 2020, we participated in the OpenMM in a collaboration that was boosted by the Chan Zuckerberg Initiative. And in 2021, now we have the chance to launch private PlayMolecule instances. So our main technical expertise is focused, as I said, on molecular dynamics simulation, software development, and machine learning. So what we do is focusing on computational chemistry at the edge with medicinal chemistry and drug discovery. This is just to give you an idea of what our tool do and how they can be used throughout the whole drug discovery pipeline. So during the early stage, so before in the preclinical trials, before getting to the preclinical trials, we can actually run ASMD interface to ATMD to discover and to characterize the targets through molecular dynamics. Then PlayMolecule can be deployed for hit finding and hit-to-lead optimization. And then furthermore, we can also trace like off-target effects thanks to pathway map and other apps. So this is a brief overview of the pharmas we are collaborating with. We have an ongoing collaboration with Janssen. We did several works published with Novartis, UCB, Pfizer, and Biogen, amongst all the other pharmas we collaborate with. So talking about PlayMolecule. So PlayMolecule in a nutshell is an integrated platform for drug discovery, and is based on machine learning algorithms and molecular simulation. So it's a graphical user interface to ASMD, and as well you can use it to visualize your workflows and what you can actually do during a drug discovery campaign or an high throughput virtual screening. So to the date, since its launch, PlayMolecule counted more than 80,000 jobs run. So it actually had a huge boost in the latest year because we constantly strive to the improvement of the platform. So it has been validated by several pharmas. We also won the Drug 3R challenge, which was a context based on the prediction of docking poses of unknown sites. And furthermore, we drastically improved the time required for hit finding to the hit-to-lead optimization based on methods that are that can be related to FEP and also a robust ligand analysis and screening. This is the benchmark of ASMD. So you can see that we are actually we can perform on the DHFR benchmark up to 1.6 microseconds per day. So we are constantly improving the performances of ASMD, and we are actually partners to NVIDIA. And thanks to their support, we are also able to improve our algorithms. So talking to PlayMolecule, PlayMolecule is a container of apps. So it's based on a web GUI, and inside this web graphical user interface, you can find all these apps, which I clustered here according to their scope. So we have MD-based simulations apps, which include Protein Prepare, which is an app that can that allows the user to prepare its structure starting from a PDB or the protein and ligand complex, and then protonate it, generate the protonation states, and adding the hydrogens. So prepare the system for eventually System Builder, that is our app that prepares the system for the simulation. So it parameterizes it, the structures inside, it adds the water, it prepares the box with the solute, and the whole soup is then run through our app that is called SimpleRun, that is the AMD interface to AMD, the engine for calculation. And besides this, when you have a protein ligand complex, you will need to generate the parameters for the ligand, and this can be done through our app named Parameterization. And furthermore, if you don't have a strictly globular system and you have a membrane protein such as GPCR, you can prepare a membrane using Membrane Builder. So after the run, you can use PlexView to see the 2D interaction throughout the simulation. And then we have a special kind of simulation that is called adaptive sampling, that is based on Markov state model generation, but this is an advanced feature of PlayMolecule. So this is what you can run on the public server, except for adaptive sampling, which may be asked on a private instance. So then for the output virtual screening, we have our app that is targeted for docking, which is Calidoc. BindScope, that is an app based on machine learning, and docking, which will predict the binding or not binding, of the ligands. Then we have KDeep, that is a predictor of KDeep, that is a predictor of the binding affinity, so the KD of the ligand to our complex. And then delta delta, which is based on the improvement, which is a heat to lead optimization program, and is based on FEP, something that is looks like FEP. And then pathway map, that allows the user to predict off-target effects and potential metabolic issues of the compounds. So from the chemical space exploration point of view, we have a ligand-based ligand and structure-based apps. So the structure-based apps are in order to identify and discover new pockets. And for this, we have Deepside, that is a machine learning predictor that uses just the structure of the protein and finds cavities or potential binding sites. And we have, besides CryptidScout, that is based on a mixed solvent MD, in which we have a probe that is a small molecule, usually a fragment-like molecule, which is used to probe the surface of the protein. So in this case, we have, beside water, that is the main solvent, we have another co-solvent, which is our probe, which can be added up to concentration of 0.25 molar. And this is in order to test eventually new allostatic pockets forming throughout this, protein moving inside the simulation. So it is in order to have a more dynamic approach toward pocket finding, because we know that the systems are not actually steady. The crystal they are not as they look in the crystal structure, but protein used to move a lot in the solution. So we need a live picture of what is going on. And then we have ligand and ligdream that may be used to suggest to the medicinal chemist new ideas for synthesis or for new compounds. And these two are based, ligand is based on generative adversarial neural network, and it uses a template which is fed to the algorithm to generate new scaffold according to some parameters set by the user. So that said, we are going to take a look at two systems today. So the first one is a classic globular protein, and in this case it's the peroxin 14 or aka PEX 14, which is the system I mainly worked during my PhD, so I know this system quite well. And in this case, the PDB code, which you can retrieve for testing purposes or just to inspect it, is 5L87. This was the acme of 3 years of early stage drug discovery, and this is the first attempt to show that actually this is a druggable target. So it can be drugged using a small molecule. And this is one of the first inhibitors developed by my group in Munich, the Popovic's group, and a first round of E2LED optimization. Then on the right, we have a membrane protein in this case, which is the 5-HT2B receptor, which is a serotonin receptor, and the PDB code is 5TVN. And this is the first X-ray structure of LSD bound to this receptor, because we know that LSD is not specific for the HT family of receptors, and this was the first ever 5-HT2B structure of the LSD bound to this receptor. So these are the ligands. On the left side, we have a pyrazole 4,3-C pyridine family of scaffold. This is one of the best lead compounds that is published in the paper that we will go just briefly through in the next slide. But this is the proof of concept of the druggability of PEX14 and the first attempt of showing that this may be used to develop new terpenocidal drugs. And on the right side, we have the very well-known LSD, and this is actually one of the most powerful psychotropic drugs. And as I said, this was the first attempt to crystallize the complex of 5-HT2B. And this is a crucial step, may look like it's not only for music production, but it may be used for the future development of new benzodiazepine-like compounds with less side effects and which may be better tolerated, and also to have a better understanding of the neurochemistry of the brain. So that said, what we are going to do is what I'm going to show you is how you can design your own workflows based on based on the available knowledge. So in this case, we are going to use compounds from this paper, which is, so the compounds will come for the virtual screening for this paper, and there are 65 analogs of the molecule that you saw of the PEX14 binders. And this will be particularly useful to show you how Scaladoc can actually do MCS, so maximum common scaffold alignment, and then perform docking in order to align all the congeneric compounds available in a library. And starting on this, building on this, I also retrieved some 2D similarity, 2D similar scaffold to the compound that you saw, and I filtered them by Tanimoto search, getting like 150 compounds from the real database that is available from Enamin at this address. So in any case, you would like to perform this again, you just have to input the smiles of the ligands of PEX14 and then filter it by similarity 0.8. So now it's time to show you the workflows. So first, when you get to www.playmolecule.org, this is the welcome screen. So this is what you will see if you land on the website. We have a workflow here which highlights all the apps that are available on the website. So we have in the target identification and validation the MD apps or the structure-based discovery apps like CryptidScout and Deepside. Then in the lead discovery, we have our at the interface actually between lead and discovery and optimization, we have Scaladoc, KDeep, and all the HTVS apps. So beside that, we also have Parameterization that is used for to generate the parameters for custom ligands in the simulations, and pathway map that can be used to test like any off-target effect. Furthermore, to display the 2D interactions, one may use PlexView, which is our equivalent of LigPlot for people that are skilled in the art, I will say. And so this is the first, the landing screen on PlayMolecule. If you scroll the page, you will get to the app, and this is just an overview, a screenshot of what you can find. So there's the app, a small description of the app, and then the get started, in which you may find more useful information on the literature behind it. And so in this case, you scroll the apps and you may test also the other one. So a very new feature of the new PlayMolecule instances is the data center. So if you decide to create an account, a private account, which you can do even signing in with Google, you can store a certain amount of data on your private cloud computing space. So in this case, you may upload your molecules or your structures, or if you have an ensemble coming from NMR screening, for example, you would like to screen all the possible conformations, then you may upload them and feed it to the software. Here, you can choose your file, add your remote path, like your local path, add some commands just for you to keep track on what you uploaded, and then the tags as well. Like if you have one target, in our case, like 5HD2B or PEX14, you may add it as a tag in order to quickly retrieve the data. So talking about the workflows, we may cluster them in three main purposes. So in this case, we have the workflow based on MD apps, which is when you have a new structure or a known structure, and you would like to see how the system evolves through time. So starting from a PDB, from an X-ray structure, or from an NMR or cryo-EM, you can prepare your system and then simulate it for a certain amount of time, and then see what happens to the protein. Maybe you can get some useful hints on the formation of allosteric pockets, or on the conformation change of some particular domains due to the presence of water, of your ligand, or and so on and so forth. So in this case, usually a workflow consists in protein prepare, which you prepare your structure, then you build the system. Eventually, you prepare it also using Parameterization. So if you need to build a membrane, so if you need to simulate a GPCR or something that is in a membrane, you can use Membrane Builder. And in this case, you can set the concentration of the membrane and the lipids that the membrane should contain, like POPC or POPG, or you can also add cholesterol in at a certain amount. You can customize it according to your like. And then after you prepare the membrane, you can call it with System Builder from a previous job, as well as the parameterized job run on the ligand if you have one. And then you can use SimpleRun to start the simulation. So the simulation will run on our local GPU cluster. So you can start it and then retrieve it when it is completed. You will have anything stored on your account. So you can create an account on PlayMolecule and then launch your jobs and then retrieve them whenever they are completed. And they will be kept on the cloud here in our servers. Of course, no one except from you can access it. And you can use still as a public user the server, but all the jobs will be available for all the users to check them. So then a particular kind of job is the one based on CryptidScout, in which you run molecular mixed solvent molecular dynamics in order to explore the potential cryptic sites that are forming throughout the system evolving in a simulation. So you can test the effect of water as well, or the interaction between water and this de-dissolvation contribution of the probe binding to a certain pocket, and so on and so forth. So this is just for you to give you an idea on how CryptidScout works. So this is your system. In this case, this is a Ras protein complexed with GDP. And this is the system with the probes added to a certain concentration. So in this case, we use the imidazopyridine, but you can pick any of the pre-computed probes that are available on PlayMolecule, even using your custom probe. So once the simulation starts, the probe will interact with the protein. So you see here, this is a simulation running of all the probes. And in this case, the van der Waals, this is what gets in the binding site. So this is will be predicted by CryptidScout, and actually this part, in this case, this probe resembles a part of the GDP, which is the natural ligand, and in this case you can see how this interacts with the binding site and how it matches almost perfectly, how it overlays almost perfectly with the same GDP part of the protein. And in this case, you can see that the system actually may be used, in this case, CryptidScout may be used also to predict and trace the binding mode of a certain probe and how the system adapts, how the binding site adapts upon binding, which is something that in crystal structure you may infer, or like a trained medchem eye or a trained structural biologist can infer what is going on, but in this case you can directly visualize what is going on. And in this case also you can use the information retrieved to getting a binding pose, a pharmacophore model, or a binding, or validate a binding hypothesis. So moving toward the screening part, we can use docking and high throughput virtual screening, and in this case we will use a target against a library or a subset of small molecules which we can prepare and then screen against them. So we have our main docking app is Scaladoc. In this case, what we can use it for is to align a congeneric series. A congeneric means some scaffold that are really that are basically 90% the same, but they just change because one metal has been switched to an halogen or to an isopropyl, or a common medchem attempt to design a QSAR library. So you can use this to align all the scaffolds and then test which will be the compound to prioritize in the design of the QSAR. So then you can use BindScope, which is similar to KDeep. We plan to integrate it actually in KDeep. And in this case, BindScope may be used on your library to have at a glance a prediction if your molecule is going to bind to the binding site or not. So BindScope is trained on is based on machine learning and is trained on pre-trained model, but you can actually use your own. So if you have like a set of molecules that you know to be binders to your protein, you can use them to train on machine learning models and then use your custom model to see if eventually other molecules are binding or not to your protein. So after you get the docking pose and the poses of your molecules, you can use KDeep to score them and to predict the affinity. So in this case, the dissociation constant, which will be outputted as PKD. And you can use this to rank the molecules and have a clue on the next step, so the prioritization of the synthesis of the compounds. And then delta delta is it can perform as KDeep, but is actually a little bit more advanced. This was so the main domain of it is to predict the delta delta free energies of a congeneric series, like a bit what Skeledoc does, but delta delta is trained on robust models and on several proteins with more than 5,000 ligands in total. And in this case, this may be a very useful tool. It's computationally more demanding, so it can be deployed in a lead optimization phase, or maybe a mature hit optimization phase, and is actually very powerful and a bit more expensive in terms of computation, so it may take longer than KDeep. KDeep is extremely fast, and also KDeep and all of these three apps, Mindscope, KDeep, and Delta Delta, may be used using custom trained models. So this means that if you have your known series of binders, you can train on that with the affinities, and then you may deploy the new screening on you can screen the new molecules using this train set. Then finally we have pathway map that can be used to predict or check eventual side effects or off-target effects of your compound. This is based on the data available from Campbell, and it may be really useful when you have to test your scaffold for metabolic liabilities. Like, for example, if it shares features that may interact with known binders of the cytochrome for 450, which is responsible, or the cytochrome family, which are responsible for 60% of the drug metabolism in the liver and in the human cells. So in this case, you may try to identify the compounds that show more issues and troubleshoot them. So like prioritize compounds that don't show this kind of interaction. And pathway map is based as well on machine learning and is quite fast and powerful from this side. So this is in order to estimate the ADME properties of your compound. And finally, we have an exploring workflow in which you have a de novo target and you don't know where to start from. So you may take a protein and screen it, like using the structure, using either according to the degree of exhaustiveness of your search. You may use Deepside and to see if there's a candidate druggable site, or maybe you can extend the known druggable site. You may try this. It takes just a couple of seconds to run. Or if you need more exhaustive data on your new target, you can use CryptidScout. And in this case, you can set your probe according to what you think it can be a good fragment or a good starting point from a hit compound, and then run it against and use it as a probe against that target. Then you may use a mix of structure-based and ligand-based approaches using Ligand, that is our virtual chemist, and or LigDream. So the difference is that in Ligand, you can start from the X-ray structure, you feed it to the system, and then the system will come up with some proposition according to how they may fit to your to the binding hypothesis or the binding site. So in this case, you will need an X-ray complex structure. And then there's LigDream, which will only feed on the SMILE string that you input and will generate new compounds according on that. So that said, I think that the introduction is done. I hope that everything was clear. If you have any question, feel free to ask in the Q&A section. So now I will just switch to my local PlayMolecule instance. And so please tell me if everything works fine. So just a second. Okay, so this is, as I said, the landing page of PlayMolecule. So if you write www PlayMolecule, so this is my account actually, which I used to run this demo. And here everything is clickable. So you can click here and then go get to the respective app. See, and then if we go back, actually, sorry, what? So in this case, if you click on your user, you will be redirected to the main page of your user, which contains all the jobs that you run. So let's assume that you run a membrane builder. Here you have the hyperlink to your job, and you can retrieve it at any time. So here, for example, we have the protein prepare jobs that I ran before for the 2 targets. See, everything is contained in this main page. So I recommend to create a local account for you with just adding an email address and set your password, and then you can retrieve your jobs. So here everything is contained at a glance. I already preloaded everything in and clustered them according to the aim of the workflow. So I will close these two windows. So in this case, we will start with, as I said, with protein prepare. So what I did was to upload my system, and then I asked protein prepare to prepare it at the pH of 7.4, which is the standard physiological pH, to add the counter ion. I use this as the main ligand, so this will be ignored actually, because this is EDO, is diethyl glycol, and is a cryoprotectant, so it's not actually a ligand. But in this case, I have this is my compound. So if you click on the molecule, you will actually highlight the binding site. So anything that is close to your molecule and what is going on. Also, you may show the core interaction, which in this case are perfectly matched, which are basically on, this is based on pi stacking. And then here we don't see it because I toggle off, but there's a trapped water molecule. So after you run the preparing, you may actually show what the software did. So this is based on PropK, and there's the predicted pKa for all your residues for the protonation. So in this case, this glutamate, for example, has a predicted pKa of 3.81. And so it means that at pH 7.4, it will be deprotonated. So, and even in this case, like for example, this aspartate is 1.37, so it will be deprotonated as well. Arginine, of course, is a basic residue, so the pKa is 14, is above 7, and thus it will be protonated in solution. And so you get all the information that you like from here. Actually, in this case, there's also the prediction of the nitrogen of the pyridine that may be protonated, and it is protonated. It is actually borderline, but it's protonated because we know it. And so here, if you click on view protonation diagram, so this is the threshold that you set, and this is all the predicted protonation states of your compound. So this is the results of the titration, of the virtual titration of your protein. So after that, if you're not satisfied with it, you can pre-prepare the protein or download the protonation data. You will download or you may also download the PDB with the protonated protein, the titerated protein with the protons added, and also with the hydrogens added, and most importantly with the bond orders assigned to the aromatics and things like that, which are quite crucial in simulating correctly the system. So in this case, we have a protein and a ligand. The common force field only contains the data for polymers, biological polymers. So for small molecules, you usually have to generate the parameters for running a simulation. And this is what we can do with Parameterize. So this is my Parameterize ligand. In this case, is the pyrazolopyridine. And you can see here that this is protonated because this part of the core is aliphatic, and this is a basic nitrogen, which was correctly predicted by PropK in that case. And so we can see the log of the simulation of the parameterization. This is the energies. You can download the parameters as of FRC mod if in case you would like to run it locally. So we can also take a look. So I will close this in order to keep everything more lean. So you can see in this case we have LSD, and this is the core of the pharmacophoric hypothesis. This is the indole-like part, and this is something that interacts with the negative nest of residues. And here you have, this is protonated, and all the parameters which may be downloaded in order for you to run locally. Then, if you're fine with this, we have our system prepared, our ligand prepared, and then we can feed it to system builder. So this is the output. And in this case, we have here, this is the system, which in jargon is called soup of your complex and in water and counter ions. See, this guy here is a chlorine atom, which was being added to compensate the charges, because we are using like standard physiological conditions. So we assume 0.15 molar of salt added to your solute. And in this case, everything was parameterized correctly. We can show some water bridging, and throughout the simulation, we will also see some molecules getting in this and getting trapped in this nest of hydrogen bonding partners. And the same thing can be done for the, of course, the LSD system. So in this case, I will just show you what SimpleRun will take as an input. Clicking here, you can access all your apps at a glance. So in this case, SimpleRun will be used to run the system, which you may select from here if you already prepared using System Builder, which is this app that I'm going to open now. And here you can see what the app takes as an input. So you have the PDB, the protein prepare job. You can titrate your protein. You have the parameterized ligand and the membrane builder eventually, if you like the membrane, and if your system requires a membrane, you can prepare it using membrane builder, which is what we are looking at now. So in this case, I just use POPC and POPE 50-50, which is the standard composition of a membrane you can use for MD. And I can simply select this when I'm building my system from here. I can also upload my custom membrane. And At the end, SimpleRun will run your job. In this case, I just uploaded loaded the PEX14 system. So this is the trajectory playing, as you can see. I hope that it will be smooth enough. So this is what actually happens to your complex when you simulate it for a certain amount of time. You can set the amount of time in SimpleRun. And so in this case, you will have protein contact cluster, meaning the sites that are closer to your protein. You may cluster them by RMSD, so what is moving the most throughout the simulation. But one of the most useful information that you can retrieve is this PDF report, which will tell you what is interacting where, which is the profile of the RMSD of your simulation, so if it converged or not, or whatever. And here you have tables which show, I will try to, yes, so the clusters based on RMSD, the clusters based on contacts, so how many frames you see that kind of interaction happening, and the results throughout the simulation. So you can download at any time this information. This is the RMSF, meaning the root mean square fluctuation of your ligand based on the starting frame. So how much the system moves from the starting position. And finally, the ligand plot interaction in 2D. So what are the most contributing, which is the interaction type, and who is mostly contributing to the binding. From the binding side to bind the ligand. And so you can download everything. You can actually just start PlexView, which is like the 2D plot viewer. And you can see here that we have mostly pi stacking, as we predicted before. And this is your ligand and all the possible interaction. So that said, we may go rapidly to CryptidScout. So in this case, this is the output of CryptidScout and all the pockets that were identified after the screening. So CryptidScout will run parallel jobs and then will trace the population of some states, so where the probe sits most of the time, and cluster them. So in this case, we can actually predict also the delta G of the spots. And if we click here, this is the best identified pocket according to the CryptidScout runs. So in this case, this matches exactly what has been done in the design phase of the ligand, which is simply connecting these two places using a scaffold that may also interact with these two phenylalanines. And in this case, I used as a probe the phenol, which is available as a pre-computed probe, but you can use your custom probe. See, this is the solute with all the probes pre-computed and before the run. So then the system automatically will build everything. This is the system build job with all the probes. You have a lot of water, and here you can see actually the probes. So the probes are set, are put randomly inside the soup in order to avoid like precipitation and aggregation of the probes. But this will be your final soup that you are going to simulate, and this will output this kind of results. So you will actually perfect match, in this case, the binding hypothesis, which are these two interact inside, but the system also identified something around this area. Actually, this part is then what sits at the interface with the membrane, so it makes sense that something is because this part is a part of the PPI. So this is the main PPI interacting with the external cell and actually the druggable sites, and you can connect it. So CryptidScout did a pretty nice job in this case. So I will close them in order to keep everything clean. And let's take a look at the screening apps. So in this case, I took the ligands that were available in the MedChem Journal of Medicinal Chemistry paper. and we have like around 60 compounds which share exactly the same scaffold. So what I did was simply to prepare a CSV file. So this may take some time. So what you need to do is to prepare a CSV file with a comma separated values actually, in which you have the smile string and then the name of your compound. And then you just feed this file to the YAP and wait for it to dock. You can set the tethering force, which is how much you would like the common scaffold to overlay on top of your reference ligand, which in this case will be the one that we have from 5L87. And the highest, the better, in my opinion, because in this case you will just have the alignment of all the stuff, because literally they are the same molecule. So they are just derivative of a QSAR, so Congenerics. And in this case, the software is telling me that compound number 1 is the best one, the one that should be eventually prioritized. And it's actually amongst so this is the score coming from RDoc, and the Scaladoc score is slightly higher because maybe this has a better overlapping to the starting structure. And so what I did was to run the same for the Congeneric series, and in this case I used the enamine compounds and I docked them. So what I can do here is to show, so see, we have our compounds here, the Congenerics, which of course have a better affinity. And then the first compound of the enamine series will be this Z here, this compound, which is the best compound in the series. So then what I did was to see if my enamine analogs were able to bind the structure, and I ran a quick Deepside bind scope analysis. And actually, the compound, so these are all the docking posts aligned, don't mind at the where structure, but this is like the average position of the scaffold. And actually, this is matching what we saw before with CryptidScout, one of the two pockets. It's too short to match both of them, but it may match one at a time. So actually, this may be like a good fragment to starting from. And even in this case, you can, again, show the ligands. Like if you like this one, you may actually see even this one is matching the, this is actually matching the PPI original interface of the protein, because there's a tryptophan here that is binding to the natural partner of the PEX14. And then what I did was to run either, so first I trained KDeep on the known binders, and then I just run it on the available enamine results. And of course, I mixed both the, so what I did was to mix both the subsets, and the best compounds that came out were the known compounds. But this is like from the enamine series, which looks like a good hit. And you can see that actually almost matches quite, it matches quite well the binding site and the binding hypothesis, except for maybe there's something weird happened with this Fanny ring. But anyway, I would say that this is a good hit. Of course, it's a neat compound. The other compounds come from an already optimized QSAR run. So those were correctly prioritized by KDeep. Actually, this was marked as one of the best, and this is what we observed from the experimental results. And also this one So the results are actually quite nice. So going forward, what I did was to train delta delta, which is something that predicts the delta delta, and so the difference in delta G of the compounds. And I trained this on the known ligands. So see, we have 65 trained ligands and 150, almost 150 molecules testing. And so according to, this is like the distribution of the KD and how they are connected in the multidimensional analysis of the machine learning algorithm. I won't get too technical because this is even trickier for me, that I'm just a poor medicinal chemist, but our developers are working to improve these models day by day. And actually, this is used to prioritize the selection of compounds. And so what it predicts is the pick 50, and the pick 50 minus the standard DSTD, the standard error, I think. And so you may have a clue of what compound is to be prioritized of the unknown screening hits. So those look like quite reasonable from a medchem point of view, and it looks also that they may fulfill the required binding hypothesis, the required binding hypothesis. And then finally, in the screening, you have pathway map. So here, what pathway map does is you have different metabolic pathways, and the system screens using machine learning algorithm the similarity to known ligands of these systems. So in this case, what we may hit is something that is similar to the amino acid metabolism, because we have a feature that looks like the indole in the tryptophan. So it may undergo some mutation, or some metabolic issues with that. But for the most, we see that with some pathways, for example, like cell motility, it shares almost nothing, no common features except for this. So you may actually click on this, and let's click on this metabolic pathway and zoom on it. So in this case, in the purine metabolism, we don't have any affinity. So it's something that is coming from the metabolism of amino acid. And in this case, here you will have like for these are your compounds, and this is the how they look similar to the known ligands of these protein families. So this can be used using this job was run using KEGG. That is one of the two available databases. And that said, we are almost done. I'm just going to show you what Deepside predicted here for the PEX14 target, and here it matches perfectly the binding pocket of PEX14. So Deepside is actually very powerful because it takes almost seconds to run. So it's quite useful if you don't want to spend time at having a clue. So with this, you can actually adjust the isosurface of the predicted voxels in this case, and I think that it did pretty good job in predicting this. I also ran it for the 5HD system, which is this one. In this case, it found more contacting site because this is a membrane protein, so anything will be in contact with the membrane. And so here you can switch and fine tune the isosurface. This is where LSD sits. So this is the binding site, but eventually something may bind also here because there may be something interesting happening here. This is the intracellular end of the GPCR. So yeah, Deepside is a structure based, and it may be used if you need more hints on what is going on. You can just run CryptidScout to understand if those are actually druggable pockets or just artifacts by membrane contact. And at the very last, we come to our system to generate new chemistry and to explore the chemical space in explore chemical space. So in this case, I used ligand, which is I started from the known crystal structure of PEX14, and I ask it to generate some compounds. Some of them are fragments because in this case it was tricky for the system to connect the two sides, and actually the system we had comes from some sort of fragment merging effort, and so it mainly suggested fragment-like compounds in this case, but this looks like a good hit to me. Not properly very basic one, I will say, but something you can work on. So it's just for you to have new clues on potential lead scaffolds or new chemistry or something that you may you didn't think about it. Like for example, you may also have a lot of nonsense results because this is something that no chemist will go through for parameterization, but this is like something which you may work on, or like this core, you didn't think about it, but you may use it for connecting stuff. And also this fragment is a bit weird, but as I said, you can find useful hints in what you can do. And then we have LeakDream, which is so this is my input molecule. In this case, you just need the reference ligand, and then the system will do the same that ligand did, but without using the receptor structure. And I will say that there's the margin for improvement in this case because they are biasing away from your molecule because of course you would like some degree of scaffold doping in this case. And the results look better compared to the other one, but it was a tricky target, I will say. So even in that case, it made some sort of sense. So you have like some reactive or potential metabolic issues. So, but this, except for this, may be used for or may be considered. So, like this in particular may be considered for further optimization. This one as well. This looks nice. Maybe it's a good idea. So it's a good suggestion. That said, I will just take your, if you have any questions, please feel free to ask. And I hope that it was clear and that it's useful for your job. Thank you.
Perfect. Thank you very much, Roberto. Just a quick reminder for the audience, if you have any questions, then please go ahead and write them in the questions tab so that I can read them out for you. So yeah, whilst we wait for the questions to start coming in, I actually have a couple of my own for you, Roberto.
Sure.
So the simulations you were just showing us, are they being run locally on your machine, or are those jobs being sent off to a server somewhere?
So those are have been run on our PlayMolecule servers. We have a dedicated server for that GPU cluster actually, which
Right.
can be is available for the users of PlayMolecule exclusively. So
I see. Okay. Right. So I guess my main question is PlayMolecule well integrated for high performance computing applications? I only ask because the talk from our last seminar actually covered performance optimization services for HPC. So I was just wondering whether PlayMolecule has been optimized with this in mind at all.
Yes, it's actually using, so in the local instances, it uses local queuing system, which is based on SLARM.
Okay.
And yeah, it may be used on CPU clusters, but most likely it's better to say for simulations, GPUs or machine learning training, GPUs will be the way to go because they are way faster than CPUs. But it may be deployed on CPU clusters or as well GPU clusters.
Sure, I see. Thanks for that.
You're welcome.
Okay, I'm not seeing any questions yet. So that list of available apps that you showed us, is that a complete list or is it still growing?
So we constantly improve PlayMolecule. So we think of merging some apps like BindScope and KDeep. We are currently working in doing so. So maybe we will have. So we are also working on an interpretability app of because in the machine learning models, usually you just use them as a black box, but we are developing a new app that is called Glimpse, which may help the user to visualize the connection between the data inside the models, just for you to have a clue on what data are correlated and how. So yeah, I think we are going to add and remove some apps like cut and add something here and there.
I see, right.
Yeah,
I have a question here for
yes.
you. So Roxana Maria asks, she has a question about adaptive sampling. Do you need a license to use it?
Yes, for adaptive sampling, yes. You need a private instance or to request a collaboration with us for apps. But all the other system, so all the apps that you see on this demo are available for free.
Okay, I'm still not right, so I'm not seeing anything else coming in. I've got one more for you. So I know of other systems for general molecular visualization, such as UCSF, Chimera, and PyMol. So I know these aren't specifically targeted at drug discovery, and all of the apps you showed us provide some very powerful functionality. Are there any other ways that PlayMolecule differentiates itself, perhaps?
And so in this case, we just added the Mol Star visualizer, which is the same that PDB uses. So the website of PDB, and it's quite powerful. So the structures, the output of PlayMolecule may be downloaded and open with VMD, Chimera, or PyMol as well. So according to the user needs. So PlayMolecule actually can just be used as an input-output machine. So you feed it with your input and you wait for the output and then you process it according to your needs. Yeah, I hope that this answered your question.
Yes, it has, thank you. I've got another question here. This is from Ludovico Pepito. What level of customization can we achieve through PlayMolecule in respect to the regular ASMD scripting? And will you include QMMM molecular dynamics?
So actually, we are working on some QM implementation. Parameterized already can process N1 and N2 calculation. So it actually includes some QM by default. So in this case, the scripting of ASMD will allow the user to do more fancy stuff, I would say. So PlayMolecule in that respect is just to, let's say, democratize the use of ASMD, even for users that are not very well experienced. So if you are experienced with ASMD, I will say that scripting will be the way to go. But if you just need this as I do as a medchem, like just for having a quick glance on how my system evolves through time, then I will just switch to PlayMolecule, because this is the purpose of it. So I hope that this answered.
Sure, I understand that. So here's a question from Mediora. Are all atomistic force fields supported for simulations?
So for the simulations, we support Amber, force field, and Charm so far. So like the membranes may be built in both Amber and Charm. So we are using the last version, we are using Amber 19, I guess, but the user may choose its own force field. So you can upload your parameters if you are not satisfied with those. Yeah, we're sticking to those because they are the most common one, but we are also working on the open project and the force field.
Okay, there don't seem to be any further questions coming in. And Roberta, if you wouldn't mind switching to the final slide, just as we come to a close here. So yes, unless anything is submitted in the next few seconds, I think this would be a good point to draw the session to a close. Thank you once again, Roberto, for giving us your time today and for showing us PlayMolecule. It's a really impressive tool. Looks like you can do some very powerful stuff with it. Just one final reminder that this e-seminar will be available on the CompBiomed YouTube channel and website, which is accessible via the link that is shown on screen currently. Finally, thank you to everyone in the audience for attending, and I hope that you will enjoy the rest of your day.
So thank you very much for having the chance to talk about PlayMolecule, and thank you, Tim, for the very nice words. And thank you for all the audience for the presentation, for attending the presentation.